Module 6, Part 4. So we are continuing our conversation on the extension of cell cycle, and in this segment, we will focus on cloning. Now, when we say cloning, most assume immediately that we are going to talk about gene cloning, which is manipulating genes by recombinant DNA technology and often putting genes in different organisms and produce the protein product of the gene, etc. But here, we are not going to talk about gene cloning. We are going to talk about whole cell cloning to clone animals. So why are we discussing this in the context of cell cycle? The reason being, a clear understanding of the cell cycle control and programming is required to successfully clone a cell. Although many animals have been cloned, still there is a high failure rate in this process, and we are still learning the cell cycle controls that play a role in this process. So when people talk about cloning, what they really mean is cloning or producing a whole animal. Usually they are referring to producing a whole animal by a process known as somatic cell nuclear transfer. And we already heard a little bit about this when we talked about the Nobel Prize winner. One of the individuals who shared the Nobel Prize actually pioneered the somatic cell nuclear transfer in frogs. They have been cloning frogs for a very long time, for decades. But the first mammal that was cloned was this sheep dolly. In this diagram is the process of the somatic cell nuclear transfer compared to a normal fertilization process. So as you know, during normal fertilization, you have the sperm and the egg cells come together, and you end up with a single-celled embryo that eventually develops into the blastocyst and then into a whole organism. So here is how the somatic cell nuclear transfer happens. To do this, you take an egg cell and you remove the nucleus, or you enucleated that cell. So now you have an egg cell that does not have the nucleus. And then you take a donor cell and you remove the nucleus from that adult cell and insert it into the egg. Remember, normally the egg only has half the amount of genetic material. But this nucleus that's taken from an adult mammalian cell, a diploid organism, will have the complete set of chromosomes, and you insert this into an egg, and the egg can continue on its differentiation. And again, this was pioneered in frogs, but the first mammal that was cloned was this sheep dolly. Since that time, a number of other animals have been cloned, and cows, pigs, not shown here are horses, rabbits. Over 20 different animals have been cloned. And here is a cat that was cloned, and this was known as CC, or copycat, and this cat really made the news when it was cloned. A few years ago, a company in business in California called Genetic Savings and Clone was offering services to clone your pet. There are still a couple of companies today that, for a hefty fee, will try to clone your pet. So in the case of these cloned animals, it's interesting to think about the relationship between the nuclear donor and the actual cloned animal. So here is a cat which was donated the adult cell that was used in order to clone the cat CC by somatic cell nuclear transfer. You may think that this donor cat as the mother cat, but in fact, the donor cat is a twin. You can think about the cloned animal as the late-born twin. So also shown is Snuppy, the Afghan hound. This puppy was one of the 3 puppies produced from 123 surrogate mothers, and he is the lone survivor. Also shown is a mule, Idaho Gem. As you know, mules are normally sterile. In 2017, Chinese scientists reported the cloning of 2 monkeys using the somatic cell nuclear transfer. Monkeys are primates, and since humans are in the primate family, these cloned monkeys reignited the ethical discussion concerning human cloning. Just as a side interest, a few years ago, researchers embarked on the ambitious endeavor to clone a woolly mammoth. The woolly mammoth cloning project, and so far it has been unsuccessful. Obviously, they cannot do it by somatic cell nuclear transfer from a woolly mammoth, but they are using a frozen specimen using DNA from the specimens. This has been a challenge since some of these specimens are at least 4,000 years old or older. However, a group of researchers from Harvard University are working to bring extinct woolly mammoth back to life by using the CRISPR technology, the new gene editing technology. Basically, they copy the genes from the DNA that is extracted from the frozen woolly mammoth specimens. They use the sequencing data to copy genes and insert the woolly mammoth gene sequence into the genome of an Asian elephant. It is not cloning in the traditional sense, more like a genome to resemble that of the mammoth. As far as I know, they have successfully created cells in vitro, but no embryos yet. So have any humans been cloned? This pair here owned a company known as CloneAid, and they claim to have cloned a human and brought that human to live birth, so to full development. But that turned out to be a hoax. Another in South Korea in 2004 reported that they had cloned a human in vitro, but later retracted their claim. So there is no evidence to date that a human has ever been cloned. But why would we even want to clone animals or humans? So there are a couple of reasons, at least in the case of animals. In terms of agriculture, it might make sense to clone animals. Say you have a champion livestock, a certain kind of champion line of livestock, you might want to clone these animals to propagate that kind of a business. If you have animals that have favorable phenotypes, you may want to clone them for economic reasons. In the case of woolly mammoth, Russian scientists propose to repopulate the tundra with herds of woolly mammoths, and theoretically their grazing would trample the tundra, releasing the greenhouse gases, thus reducing global warming. Kind of a far-fetched idea to say the least. In research, however, it makes a lot of sense, especially with the animal models of the disease. So if you think about this, when you are doing research with animal models of the disease, you always have a control groups and treatment groups, or control groups and experimental groups. And now you usually have to use many animals because there is a variation from animal to animal. But if your animal are all clones of each another, that you would have a better controlled experiment. You may be able to use less animals for your experiment. So what about human clones? Two purposes have been proposed. One is reproductive cloning, so live birth cloning. I mean, some people may want to clone themselves. I don't know why. Then another purpose that has been proposed is a therapeutic cloning. So let me talk about each of these. Reproductive cloning is not a good idea. It's proposed as a treatment for infertility, but cloning is extremely inefficient. So in the case of Dolly, there are 277 attempts before they actually were successful in cloning the sheep. Only 29 even made it to the blastocyst stage. So that's pretty low efficiency. And you can see that here there's an inefficiency of cloning in other animals. In one study, it took 613 cloned mice embryos before they got 5 live births, and in another, they had 2 out of 1,000. In a study on goats, they produced 3 out of 85, pigs 5 out of 72, cattle 24 out of 496, and Sisi, the only had copycat, only 1 live birth after 188 cloned embryos. And for the monkeys that were cloned a few years ago, only 2 were produced from 79 embryos. So it's a rather inefficient process, and it really doesn't make sense to use it as an infertility treatment. There's also some evidence that cloned animals may not be normal. So Dolly was cloned from a 6-year-old sheep, and what was interesting about Dolly is that she died early, and she had early onset of arthritis and cancer. So it's almost as if Dolly was older than her actual biological age. And that's actually kind of interesting, and it suggests that cloned animals may not be normal. Also, it has been observed that cloned mice have shortened lifespan compared to their non-cloned counterparts. However, concerning Dolly, there are other sheep cloned from the same genetic material as Dolly, and so far they are healthy and aging normally. So researchers are reevaluating this idea about the cloned animals not being normal and generally conclude that cloning does not perturb the aging process. All right, so what do I mean by therapeutic cloning? Therapeutic cloning is the process by which you generate embryonic stem cells to treat yourself through somatic cell nuclear transfer. So what you would do is you would take an enucleated egg and you would take some kind of somatic cell from your own body. And you would remove the nucleus and you would place that nucleus in this enucleated egg. You would bring that cells or a group of cells to the blastocyst stage, isolate the inner cell mass, and culture these embryonic stem cells. Then you can differentiate them into whatever you need for whatever your ailment you have: blood cells, nerve cells, heart cells, liver cells. So say if you have some kind of degenerative disease in the muscles or the nerves, you might manipulate the genes in these cells to overcome certain malady, and then you would transplant these back into the patient. That's what is meant by therapeutic cloning. In this case, you eliminate the disadvantage of transplant rejection, which is one of the disadvantages of embryonic stem cells. But you definitely are still faced with some ethical issues of destroying the blastocyst to generate these embryonic stem cells. All right, and as I mentioned at the beginning, stem cells and cloning are controversial issues in modern day science, and we just don't have the time in this course to delve into the bioethics. There are many college courses dedicated to studying this topic. But here are some of the questions that arise in addition to some of the questions I proposed at the beginning. What is the moral status of a human embryo? Scientifically, the embryo is a human being, but is the embryo really a person, or is it a piece of property? Who owns embryos? Currently, there is a surplus of embryos that are frozen from infertility interventions. And the question is, how do you store these? How long do you store these? And actually, who owns these embryos, or does anyone own them? And then you have to ask questions about scientific freedom and scientific stewardship. Should experiments be conducted just because you can? Of course, every age, every generation is faced with that question by some other kind of scientific breakthrough. And these are really interesting questions that are worthy of thought and worthy of discussion. So this concludes part 4 of module 6 and also completes our module 6 discussion.